Good afternoon and namaste. I am Dr. Sutripta Sarkar of the Department of Food and Nutrition, Barakpur Rashtraguru Surendranath College, Barakpur, and today I will be delivering a lecture on vaccines, its importance, types and strategies. So, this particular topic is a part of the course BSc Honours in Food and Nutrition of the West Bengal State University. The paper is FNTA Core 12T, and this is a very important topic, especially with the outbreak of pandemic. We have all been waiting eagerly for the vaccine from the beginning of the pandemic, like we were waiting for the vaccine to come. We almost know everything about vaccine, barring I think a few facts. But as students, we need to understand vaccines, its importance, its utility, the type of strategies that are taken, that are adopted to make and create such vaccines. So, let me start the lecture. As we talk about vaccines, the first name that comes in our mind is that of Dr. Edward Jenner, the British physicist who is credited for creating the first vaccine. Smallpox was very prevalent during that time, and I am talking about the late 17th century. The late 17th century, actually, Dr. Edward Jenner, what he did was that he used pus cells from a milkmaid having cow pox. So, there were these rashes and pus oozing out, and he took those pus cells and he successfully used it for the treatment of smallpox. This is how the first vaccine was discovered. Now, this is the flow chart of vaccine over the years. So, it was in 1796 that smallpox vaccine was first discovered, and that is why Edward Jenner is known as the father of immunology. By the time 1880s came, Louis Pasteur was doing a lot of research in vaccines. He also developed and coined the term vaccine. He developed vaccines for rabies and cholera. Early 1900, the toxoid vaccines were used, especially for tetanus and diphtheria. Polio vaccine was discovered in 1950s, and the Sabin and Salk vaccine, or the oral polio vaccine, which is very much prevalent, is a result of those researches done. Haemophilus influenzae, the vaccine was discovered in 1985, hepatitis A 1992, rotavirus 1998 was first approved and licensed in 1998. However, there were a few shortfalls in the vaccine. So, it was taken off the market, and later on a safer vaccine against rotavirus was licensed in 2006. Now, in 2006, we also had the first HPV vaccine, human papilloma virus vaccine, and it became available in 2006 itself. And in 2019, the Ebola, after the Ebola outbreak, we had the Ebola vaccine, which is found to be 95 to 100% effective against the Ebola virus. Okay. And finally, in 2020, the later part of 2020, Pfizer came up with the vaccine, COVID-19 vaccine, which was approved, and as of now there are several COVID-19 vaccines which are available. Also during this 2020 and 2021, it is not mentioned in this flow chart, but we know that we also had the first RNA virus vaccine, which is we had a vaccine for HIV. Now, we are talking about vaccines, but what is essentially a vaccine? Basically, vaccines are biological preparations that provide active acquired immunity to a particular infectious disease. It contains an agent that resembles a disease causing organism and is often made from weakened or killed forms of microbes, its toxin or one of its surface protein. So, this is vaccine. And what about immunization? Immunization is actually the process that elicits a long lived state of protective immunity against a disease causing pathogen. Okay? So, exposure to live pathogen followed by recovery is the root of immunization. So, if you are exposed to a live pathogen, supposing that you were infected with the COVID-19 virus and then you recovered from it. So, therefore, you have got some immunity against that particular strain of the virus. Okay? So, vaccination is an intentional exposure to forms of pathogen that do not cause the disease. So, in vaccination, please remember, vaccines are biological preparations that provide active acquired immunity to a particular infectious disease. Okay, vaccines contain an agent that resemble a disease causing microorganism and is often made from weakened or killed forms of microbes. It is toxin or one of its surface proteins. Okay, so this is vaccine. What is immunization then? Immunization is actually the process of eliciting a long lived state of protective immunity against a disease causing pathogen. Exposure to live pathogen followed by recovery is the route to immunization. So, if supposing you contracted the COVID-19 vaccine, you contracted the COVID-19 virus and you got the disease and you suffered and then you later on recovered. So, that should elicit a long lived immunity against that particular strain of the virus. Okay. So, vaccination is actually an intentional exposure to forms of pathogen that do not cause the disease. You have to remember that in vaccine preparation, we are actually taking a dead part of the vaccine or some DNA or RNA or maybe polysaccharide or maybe some other part of the virus, and it's only a part of the virus or it's a form, a non-pathogenic form. Okay. So, that will elicit some response in your body. It will not exactly cause the disease, okay, but the response it is caused, that will generate antibodies, that will generate memory T cells, and that will give you a long lasting immunity against the disease. So, before we get into the detail of vaccines, there are certain terminologies which we have to understand. First is passive immunity and active immunity. So, when we start studying immunity, we know that there are two types of immunity, basically how they are acquired. Based on that, they are differentiated into two parts, that is a passive immunity and active immunity. Passive immunity is comes through natural maternal antibody, IgA, that it can cross the placental barrier and it goes into the baby IgA, and then there are immunoglobulins, okay, the antibodies and the antitoxins, okay, all these give passive immunity. What is active immunity? Active immunity is when you have contracted or when the virus or bacteria or whatever organism has entered your body, you have suffered, you have got the disease and you have recovered. Okay? So, that is active immunity. Active immunity can come either from infection, you getting infected, or you getting vaccinated. Okay? So, vaccines can be made from different types of materials. It can be attenuated organisms, inactivated organisms, purified microbial macromolecules, cloned microbial antigens, and multivalent complexes and toxoids. So, passive immunization, it can occur naturally via transfer of maternal antibodies across placenta to fetus, as I already mentioned earlier. Injection with preformed antibodies, human to animal antibodies can be used. Injection of animals antibodies prevalent before vaccines. Okay. So, what happens here is, in case of tetanus, if I take the example of tetanus, whenever you get a cut or something by iron or any sharp object, you will be taken to the doctor to get the anti-tetanus injection. Basically this tetanus, the bacteria which causes tetanus is injected into an animal, maybe horse, okay, and that horse produces antibodies against it. That antibodies is collected from the serum, horse's serum, it is purified and it is injected into you. Okay. So, you are getting preformed antibodies. The tetanus organism is not attacking you or attacking your body or your cells directly, but you are getting preformed antibodies just in case that the tetanus organism, the organism causing tetanus comes in or enters your body. So, there are antibodies ready to fight it off. Okay. So, passive immunization, the effects of passive immunization are temporary, hence every time you get a cut, every time you have to go at least, I mean there is a time period, there is time period of within 6 months. If you have taken the anti-tetanus injection, you need not go, but beyond that you have to again go for the injection. Okay. That is because the antibodies have a limited lifespan. So, common agents that are used for passive immunization, for black widow spider bite, horse antivenin, botulism, horse antitoxin, diphtheria, horse antitoxin, hepatitis A and B that uses pooled human immune gamma globulin. Okay. And for measles also it is the same, pooled human immune gamma globulin. Rabies also we use gamma globulin, human gamma globulin. Snake bite is, there is also the horse antivenin, and for tetanus pooled human immune gamma globulin or horse antitoxin, both can be used. Okay. So, the transfer of antibody will not trigger the human immune system. As you can see, you are already injecting preformed antibodies. So, they are already formed, they just need to, if the causative agent for the disease comes, they need to fight back. They you don't need, the immune system as such is not triggered. Okay. There is no presence of memory cells. Memory cells are not formed in passive immunity. Okay. Achha, risks that are included in passive immunization is that recognition of immunoglobulin epitope by self immunoglobulin paratopes. Some individuals produce IgE molecules specific for passive antibody leading to mast cell degranulation. That is, sometimes these antibodies which are coming from the horse, our body may recognize them as foreign. Okay. So, we might develop a allergic reaction to that. Okay. The IgE gets stimulated and the mast cells degranulate, and you might get a severe allergic reaction to that. Okay. So, that is a risk or danger per se in case of using passive immunization. Some individuals produce IgG or IgM molecule specific for passive antibody leading to hypersensitive reaction, as I already mentioned that against the antibody that has been injected, since that is prepared in a separate animal body or a foreign body, that is can be recognized as foreign by your own body. So, you may get hypersensitive reactions. Now, active immunization, natural infection with microorganisms or artificial acquisition, that is vaccine. Okay? So, you can fall sick, the organism or the causative agent that entered your body, you got the infection and you fall sick, that and then you recover. That is a form of active immunization. Okay? And also when you take vaccines, okay, artificial acquisition is vaccines. Both stimulate the proliferation of T cells and B cells. B cells generate antibodies, we know. T cells will be, there will be natural killer T cells and memory T cells. Okay? That memory T cell formation is very important for a long lasting permanent impact of vaccination. So, here we see this figure shows that immunological memory versus serum antibody levels. So, serum antibody level, you can see that it is almost dipping, the level is almost dipping to half in 6 months. But whereas if your immunological memory has been activated, memory T cells have been formed, they continue forming and they remain quite active for more than over a year, sometimes lifelong also. Okay? So, what is the importance of vaccination? Why is it so important to get vaccinated? Why there were so many advertisements and so much propaganda during the pandemic that please, the vaccines are available, get vaccinated, get vaccinated. Why is it so important to get vaccinated? Okay? Because vaccines prevent the spread of contagious disease, and hence vaccination is very important for family and public health. Okay? So, vaccination, it also protects children from serious illness, not only children but also senior citizens who are immunologically compromised. They also, there is a big role that vaccine plays in protecting them against diseases as well. Vaccination prevents outbreak of diseases. So, you can see that after the COVID first phase, then second phase we had the Delta, third phase we had the Omicron, and then there were small outbreaks, mostly locally, but they could be, mostly the outbreaks are under control now, as of now, and even if it is there, it is not spreading that much. Okay? So, vaccination, it helps in prevention of outbreak of diseases. All vaccines undergo careful and long trials to ensure their effectiveness and safety. Vaccines, it is not that somebody discovers a vaccine and it's marketed, it's comes into the market and you are just injected the vaccine. It takes a lot of time, a lot of struggle, and a lot of it has to undergo a lot of tests before it is actually applied, or which it is actually sent for public use. Okay? So, vaccines when made, they first have to clear the animal models, they have to be successful in animal models, then they have to undergo the 3-phase trial, clinical trials. Okay? Phase 1, phase 2 and phase 3, and only then can it get a safety nod so that it can be applied to general or injected to general public. Vaccination also helps in stopping of transmission of diseases. So, definitely we know that now we were 2 years we were at home. Okay? We were doing everything online, and finally when we had our 2 doses of vaccine and probably also 1 dose of the booster dose, the transmission of the disease has been stopped to a great extent. Okay? Okay, so let us talk about the effectiveness of vaccines. So, we can see that how effective a vaccine can be. This is when let us take the case of smallpox. Okay. It was very prevalent in 1950s and 60s, but as soon as vaccination for smallpox became mandatory, within a decade the number of cases saw a sharp decline, very sharp decline, and now, as of now, WHO has declared that smallpox has been eliminated. Okay. Here are the effectiveness of other vaccines like rubella. You can see orange is without vaccination, the situation that was there in 1969, and rubella cases are very much reduced. Paralytic polio, 1952 it was very prevalent, over 10,000 cases were reported, but currently paralytic polio is also on the way out because of the big immunization programs that have been undertaken by the United Nations, UNICEF, and also Government of India. It is on the way out. Okay. Then in 1934, pertussis was a much feared disease, and now pertussis cases have also drastically reduced. So is mumps, measles and diphtheria. Diphtheria was like a killer disease for children in the 1920s, 30s and 40s, but the vaccine has really helped to reduce cases of diphtheria. So, vaccine strategies. What are the advantages and what are the challenges facing vaccines? Okay. So, key factors for development of successful vaccine. The first and foremost key factor is the vaccine should be absolutely safe. If the vaccine is giving some long term or short term side effects, then it should be decided by the authorities whether at all it can be used or it has to be rejected. Okay? So, the vaccine has to be absolutely safe for human use. It must be effective in preventing infection. Then the strategy should be achievable given the population in question. Okay, whatever vaccine you develop, it should be able to reach to the common people. Okay, unless you are not making it, you know, economically viable or not viable for a certain type or population of people, it will not stand. Okay. So, it should be able to generate long term memory. So, the one key factor for successful vaccine is safety, and the second one I would rate that it is long memory. The vaccine should be such that at the lowest number of possible doses, it should be able to give maximum immunity, okay, and a long term immunity. So, types of vaccines, which are administered to humans. So, on the left we have disease or pathogens, type of vaccines. The first section is showing whole organisms. Whole organisms can be inactivated whole organisms or attenuated, live attenuated organisms. Okay. So, bacterial cells like anthrax, cholera, pertussis and plague, for that mostly inactivated whole organisms are used. For tuberculosis and typhoid, live attenuated BCG for tuberculosis and live attenuated whole organism cells for typhoid. For viral particles, hepatitis A and influenza, inactivated viral particles are used. Measles, mumps, polio. Okay? Polio, the one discovered by Sabin, that uses live attenuated, and polio vaccine by Salk is actually using inactivated whole organisms. Okay? and rotavirus, rubella, varicella, that is varicella zoster, that is which causes chicken pox, and yellow fever, those are live attenuated, only the rubella is inactivated. Then besides whole organisms, so varicella zoster, which causes chicken pox, that vaccine uses live attenuated whole organisms, and so does yellow fever, that also uses live attenuated organisms, whole organisms. Okay. So, besides whole organisms, there are several other vaccine strategies. Another uses purified macromolecules such as toxins. Okay. So, in case of diphtheria and tetanus, the toxoids, inactivated exotoxin is used in preparation of the vaccine. For Haemophilus influenzae type B, which causes meningitis, that uses polysaccharide and capsular polysaccharide and a protein carrier. Okay, and the polysaccharide, 23 distinct capsular polysaccharides are used for meningitis and pneumonia. Then surface antigens are used in hepatitis B. You must be knowing that hepatitis B is the first injection that is given to a newborn baby, and for this recombinant surface antigens, okay, that is used for preparation of the vaccine. So, childhood vaccines. The 7 major childhood vaccines are HPV, that is hepatitis B, DTP, IPV for polio, MMR, HIV, that is Haemophilus influenzae type B, VAR, that is varicella, and PCV. Okay. These are the mandatory childhood vaccine as recommended by American Academics of Pediatrics. So, adult vaccines for those living in very close or which have compromised immunity, they need HIB for meningitis, PCV for pneumonia, they need a regular vaccine for influenza, all adults or elderly rather, they need this influenza vaccine. And for travelers to endemic areas, you need vaccines for cholera, typhus, typhoid, hepatitis, meningitis, yellow fever, polio and anthrax. So, anybody who is like planning to go for a jungle safari in Kenya, it is mandatory for them to visit there, it is mandatory to take the yellow fever vaccine, okay, before you go to Kenya. So, whole organism vaccines, attenuated virus and bacteria. Okay. So, what is attenuation? I have been for the past few slides, you must be hearing this term attenuated and attenuation. Okay. It is the cell which has been inactivated, okay, which has been inactivated so that it cannot cause the disease, but it can still grow to a certain degree when inoculated in the host. Okay. So, such a type, when you continue growing certain pathogenic strains of viruses or bacteria, for several years or maybe in several artificial medium. So, they then lose their pathogenicity. That is how BCG vaccine was discovered. BCG vaccine took 13 years, for 13 years it was grown on the artificial medium, and then finally they used the attenuated strains to develop the vaccine. Okay. So, attenuated are live but inactive strains. The positives is that it provides prolonged exposure to the epitopes of the organism. Okay, prolonged exposure to the epitopes of the organisms, so that it can generate a very long term memory. okay, high memory and high immunogenicity. Okay. So, attenuated organisms will require lesser number of shots or lesser number of booster doses, okay, and then it stimulates host mediated response. And the negatives are that there is a small, very very minuscule chance that that the attenuated strain might revert back to become virulent. Okay, though it is there are sufficient number of trials which are taken before the vaccine is finally approved, but there is a slight, I can say a very minuscule possibility of reversion to virulent form and certain side effects. Okay. Example, polio and measles. So, off and on you will hear reports that in some areas some children were administered the polio vaccine and they got polio, very long time back such reports were there. But in recent times, paralytic polio, there has been absolutely no reports. Okay. So, polio is also on its way out. The inactivated viruses and bacteria. Inactivated is basically you are killing the organism, okay. You are killing it by heat or chemicals such as formaldehyde, alkylating agents. Usually such inactivated virus or bacteria prepared vaccines, they will require repeated booster doses, okay. And predominantly humoral immunity, you will get humoral immunity predominantly. And there is, although you are inactivating it or killing it, there might be a small minuscule chance of containing some active pathogen. Okay. So, here is the comparison between attenuated, live and inactivated killed vaccine. So, in production, attenuated vaccine, selection of a avirulent organism, virulent pathogen or virulent pathogen is grown under adverse culture condition or prolonged passage of a virulent human pathogen through different hosts. What happens is when you grow it for a prolonged time in adverse conditions or it undergoes several different types of hosts, what happens is it loses its pathogenicity, okay, and that is when it can be used for preparation of the vaccines. Virulent pathogen is inactivated by chemical or radiation with irradiation with gamma rays, okay, in case of inactivated vaccine. Booster requirement for attenuated vaccines, generally a single booster or no booster is required, but for inactivated vaccines it will require multiple boosters. Okay. Coming to stability part, attenuated vaccines are less stable and inactive vaccines are more stable. Therefore, it is more advantageous for the third world countries where keeping everything refrigerated, all medicines refrigerated is a huge infrastructural problem. So, for them the third world countries, inactivated vaccine is the best bet. Type of immunity induced, attenuated vaccines induce both humoral and cell mediated immunity. Okay. So, humoral immunity is production of antibodies, cell immunity is production of natural killer T cells and memory T cells. But inactivated vaccines, they produce mainly humoral immunity. Reversion tendency, the chances of the organism getting reverted to the virulent form is higher in case of attenuated vaccine as compared to inactivated vaccine. So, parts of part purified molecules, capsular polysaccharide, they utilize the antiphagocytic mechanism vaccines using capsular component stimulate the Ig production, that is immunoglobulin production. Okay? Vaccines for Streptococcus pneumoniae and meningitis, purified polysaccharide which is injected subcutaneously to activate memory B cells and IgA response. Okay. It can invoke the activation if polysaccharide is added to protein carrier. Then recombinant microorganism antigen, hepatitis B, as I said earlier, that it uses the surface antigen which is cloned in yeast. Okay, it is cloned, the surface antigen is cloned in yeast, and then it is used for preparation of vaccine, may be able to produce vaccine, large amounts of vaccine this way. Okay. And also in purified macromolecules, it avoids the risk of whole organism vaccine. Okay. There is a risk of the whole organism getting virulent, that is completely eliminated here. So, 3 forms that are used are inactivated exotoxins, capsular polysaccharides, and recombinant microorganism antigens. So, inactivated exotoxins or toxoids. The toxoids, the toxin which is inactivated is known as toxoid. Okay. So, they purify exotoxin, treat with formaldehyde, produce antitoxin immunoglobulin which bind to the toxin. The exotoxin gene can be also cloned. It is another strategy. The exotoxin gene can be cloned, recombined in cell to produce large quantities of the vaccine. Such strategies are used for the development of vaccines for diphtheria and tetanus. Okay. A recombinant vector vaccine. In this, a recombinant plasmid is created which contains the gene from the pathogen. Okay. And that is transfected to certain tissues which also contains, you know, vaccinia virus that is also infecting it, and they in the tissue culture cells a homologous recombination occurs, and we develop or we get a recombinant vaccinia vector vaccine. Okay. So, which can be administered to the subjects. So, vectors that can be used can be vaccinia, polio, adenoviruses, salmonella, BCG strain of Mycobacterium bovis, and oral streptococcus. Other vectors may prove it to be safer as well. So, now in the current scenario, we see that besides all these, there are also DNA vaccines and RNA vaccines which are available. So, how is a DNA vaccine created? Okay. So, in a DNA vaccine, a plasmid DNA is taken to which an antigen gene is incorporated. Okay. That is inoculated or transfected into a host cell, and because of that antigen gene, a lot of antigen or protein expression takes place, okay, in the host, and these antigen they help in eliciting immunity to the host. So, DNA vaccine, there are very few DNA, I mean as of now only Zydus Cadila, I think they have successfully developed a DNA vaccine for the COVID-19, but it is yet to hit the market. So, their vaccine is prepared, is a DNA vaccine. And let us talk about RNA vaccine. We are talking about DNA, and let us talk about RNA vaccine. RNA vaccine, two strategies can be applied. One is through non-replicating mRNA construct, another is self-replicating or self-amplifying mRNA construct. This self-amplifying mRNA construct or SAM, it contains a genetic replication machinery from a positive stranded mRNA virus. That is how it is different from NRM. Okay? Else both are same, and they are incorporated via lipid nanoparticles into the cell. Okay? They are transfected into the cell, and as you can see in the diagram, they enter into the cell, and the mRNA they express themselves, they make protein, and the protein then they finally they take their shapes, and they take their structure. Okay, transmembrane, it can be transmembrane or it can be intracellular. Okay, and they are shown, or they are represented on the surface of the cell. Now, once the immune cells detect these proteins, a immune response is created or elicited in the body. Okay? That is how we develop immunity, by using RNA vaccines. So, the Pfizer vaccine is the RNA vaccine, okay, is a RNA vaccine. Pfizer vaccine for COVID-19, that is a RNA vaccine. Now, we are talking about vaccine, and we are just about to move out from the pandemic, and we should be talking about the COVID-19 vaccine strategies, how and what they were used. Okay? So, viral vectors were used because uses of harmless viruses. Okay? Some viruses which do not cause any disease in humans.